program between the University of Florida IFAS Extension and Martin County and is funded by Martin County. And it's an adult education stormwater education program for Martin County residents. And we try to focus on water quality related topics that are of interest to both Martin County and Southeast Florida residents. So you can see the remainder of our 2021 Virtual Water Ambassador Program series in the right-hand side. And Vincent and I will put the registration links for the next upcoming webinars. So just a reminder that you all are going to be muted right now, and we ask that you stay muted and that you stop your, don't start your video. This is for bandwidth reasons. You tend to have better connection if you don't have your video going. You can click the participant icon on the bottom of your screen to see who's joined us today. And we ask that you use the chat box for any discussion or questions, and we will hold questions until the end of the presentation. And if you need to leave the webinar at any time, just click on the red Leave Meeting button. For best viewing, we suggest that you use the side-by-side mode. This allows you to see both the presenter and the presentation slides at the same time and scale it for the size of your screen. We are recording this, or we should be recording. If we're not already, we will be. And all of these recordings are available online at the Martin County Sea Grant Extension website. They're listed by date and topic. And this is a really long URL, so just Google Martin County Sea Grant and you'll find all of the Water Ambassador recordings on the right-hand side of the screen. Just that last reminder again to hold your questions and type them into the chat feature, which we'll discuss at the end of the presentation. And with that, I am really pleased to present our speaker today, Dr. Joshua Voss. He's going to be presenting on protecting Florida's coral reefs, water quality, and coral health, which I think is really pertinent to the Water Ambassador Program and maybe oftentimes a topic that we don't get to discuss frequently enough. So Dr. Joshua Voss is an associate research professor at FAU's Harbor Branch Oceanographic Institute and the executive director of NOAA's Cooperative Institute for Ocean Exploration, Research, and Technology. His primary areas of interest include shallow and mesophotic coral reef ecology, coral health and disease, molecular ecology, marine conservation and management. Through Harbor Branch's Robertson Coral Reef Program and the NOAA Cooperative Institute, he works to discover, characterize, and protect coral reef ecosystems. Josh is a certified technical diver and scuba instructor who's completed over 1,500 scientific dives and has led more than 35 expeditions. He also teaches undergraduate courses at the Harbor Branch Semester by the Sea program and the Florida Institute of Oceanography Marine Field Studies program, graduate courses, and molecular workshops for high school students, and serves on various committees, including South Atlantic Fisheries Management Council's Coral Reef Advisory Panel, the Southeast Florida Coral Reef Initiative team, and is co-lead of their Technical Advisory Committee, of the Florida Keys National Marine Sanctuaries Technical Advisory Committee, the South Florida Marine Research Hub, and FAU's Diving Boating Safety Committee. I have no idea how Josh has time to do anything else. But after growing up in the beaches of Central Florida, Josh attended Elon University at North Carolina and earned his PhD at FIU in Miami and was a member of marine science faculty at Eckerd College before joining FAU Harbor Branch. So thank you so much, Josh, for presenting to the Water Ambassadors today, and I will turn it over to you.
Thank you, Lisa. Thank you, Vincent. And had I known you were going to read all of that out, I would have sent you a much shorter bio. I apologize. All right, let me get rolling here. And please let me know if everyone can see the screen okay.
Yes, it looks great.
Very good. Well, thank you all again. I appreciate both the invitation and your time today to learn a bit about coral reefs in Southeast Florida and how water quality can affect or impact them. I've been at Harbor Branch since 2007. and really initially worked more in the Bahamas, wider Caribbean, and some work in the Keys. And it wasn't until about 2010 that I really got interested in reefs farther up the coast into Martin County. And there's been a couple folks that have really been instrumental in that. Jeff Beal, who used to be with FWC and is now with Ducks Unlimited, and Kathy Fitzpatrick with Martin County have been two champions, advocates, coaches, mentors, teammates, all of the above. So I really appreciate their help for all things that are Martin County related. The work I'm going to present today is a hugely collaborative effort. I've got a fantastic team of students and technicians that are all in my group at FAU Harbor Branch. The folks that have contributed kind of most to some of the things you're going to see today are Aaron Schilling, Alexis Sturm, and Michael Studevin, who are all who are all students of mine. Aaron recently completed her master's and is now our lab manager. And Mike recently finished his postdoc and has since moved on to CMIS down in Miami. And then all the other folks there listed with asterisks are previous grad students that contributed to some of this work as well. We also have a great team at our marina. Jimmy Nelson and Matt Roy help us out quite a bit. And then other faculty at Harbor Branch like Dennis Anasak and John Reed have really been important collaborators on a lot of our Florida work as well. And then as we've gotten more and more involved with DEP and other collaborators from NOAA, Smithsonian, FWC in Southeast Florida, it's been really kind of refreshing to see how easily and willing people are to help one another to tackle really difficult science and policy questions. So to the folks at DEP like Christy Kerrigan, Joanna Walzak, and Jamie Monty, and again to Kathy and Andy and Scott at St. Lucie Inlet State Park have been really important teammates on the work I'm going to show you today. Most of this work has been funded by a combination of both state and federal funding. That state funding has come primarily from DEP, and the federal funding has come from a variety of resources, NOAA through the Ocean Exploration and Research Office, through Sea Grant, and through EPA. And then we also had a philanthropic donation from Joe and Jennifer Duke through the River Branch Foundation early on that helped establish our program. So when we think of these coral reef habitats, most of us have something like this in our minds. These amazingly rugose, lots of nooks and crannies, biodiverse areas, lots of life on the bottom, most of it covered by corals, supportive fish communities, and just an interesting, productive environment where lots of energy is being taken from the sun and converted into fixed carbon energy that can feed these enormous food webs and food chains. And most shallow reefs in the Keys and throughout the wider Caribbean look something like this. As we start to go down a little bit deeper, we start to see reefs where as light becomes more limiting, corals try to spread out more to get as much of that light as they can. So we see terraces like this down at depths of say 120 feet or so and beyond. Unfortunately, over the past roughly 40 years or so in the tropical Western Atlantic, many reefs are starting to look more and more like this. So coral cover being lost to various different threats, and in many cases, coral cover being replaced by soft corals or other things that don't necessarily calcify. The other main issue we tend to see in certain places is an excess of macroalgal cover. And it's particularly true in areas where herbivory has been depressed through different cascading effects through the food chain. It can also be impacted by nutrient runoff in certain areas as well. So we have both bottom down and top up effects affecting what's going on in coral reefs, and then overlying factors as well that can drive these changes. So we know these changes are taking place, and it's of concern to resource managers, scientists, and anyone that relies on coral reefs. In Florida, that's pretty much everyone in Southeast Florida. Our entire coastline is protected by reef that if we didn't have that reef, our insurance premiums for hurricanes would all be quite a bit higher. So understanding what's driving these changes and then how we can potentially manage coral reefs to start to shift them back towards high coral, high stability communities is really important. And a lot of this success that we see, what drives corals dominating space and building big habitat, is driven by the symbioses that they share with algae. So if you think of corals, remember corals are an animal. They're a cnidarian, which means they're essentially a colony of small little anemone-like polyps that grow and produce calcium carbonate skeleton that give rise to reefs, literally. The reefs accrete and grow over time. And so inside most corals, there are symbiotic microalgae or zooxanthellae, and the corals tend to provide nutrients in the form of nitrogen and phosphorus to those and protection from predation. And in turn, those algae tend to be incredibly productive and leaky with the sugars they create through photosynthesis. So those carbohydrates can then be used by the coral for growth and reproduction. So really this symbiosis drives this enhanced coral growth. And this is a simplified view of the symbiosis. There's several other members of bacteria, cyanobacteria, viruses that are all involved as well. But it's really this primary symbiosis that drives that productivity and drives the success of coral reefs. And so that success tends to result in high productivity. And the kind of productivity we tend to care about is productivity that ends up in fish. So cascading up in the food web until it gets to fish stocks that are of economic importance. But the other interesting part of the variety on reefs is that because there's lots of different niche space and lots of competition, you tend to see coral reefs as very high centers of biodiversity. So some estimates are that they house about 25 to 40% of the known biodiversity of the ocean. I mentioned this previously, but again, they're incredibly important for abating wave energy and providing coastal protection. And throughout the world, but particularly here in Florida, they drive a really important tourism industry. So for example, we have individual reefs in the Florida Keys that have over 1 million visitors a year. And we have a huge recreational fishing community that travels to Florida either frequently, annually, or just once in their lifetime to come to some of the best sport fishing in the entire world. The other aspect that is interesting about coral reefs is because there's so much competition for space on the bottom, and because many of those benthic organisms can't get up and move, there's been a prolonged history of evolutionary chemical warfare between corals and other organisms like tunicates and sponges, trying to overgrow one another and compete for space on the bottom. When benthic organisms are competing with one another, they usually are more likely to evolve secondary metabolites for defense or aggression. And those secondary metabolites have been shown to be potent for pharmaceuticals in several different instances. So things that are successful against certain kinds of cancer, things that are successful against malaria. And there's a whole separate industry and research area trying to understand what pharmaceuticals we can use from coral reefs. So taken together, coral reefs are incredibly valuable. The current estimate of net worth globally is about $13 trillion. And here in Florida, it's estimated at somewhere between 8 and 15 billion per year. And coral reefs directly support about 70,000 jobs in our state. And I would argue that probably all of these numbers are on the low side of what is real. There has not been a good systematic review of coral economic value that's very recent. And so my guess would be an updated economic analysis of corals, particularly in South Florida, would reveal that they might be even worth more. So we have this amazing reef that's highly productive, highly diverse, and really economically important. Unfortunately, there are several threats to our coral reefs. So some of those are natural, things like hurricanes, although they may be increasing in intensity and frequency as a result of climate change. Other threats come from beach management practices, beach renourishment projects that are not managed well, or dredging projects that are not managed well. ship groundings, anchoring, overfishing, freshwater discharge, and disease. And I'm really going to focus on these last two today, the potential impacts of freshwater influence and nutrient pollution in Southeast Florida, and the impacts of disease and associated coral death in Southeast Florida. So our approach to this overall is really to combine three different areas. We do a lot of exploration and monitoring. Surveys to understand what resources are available, and the exploration part of it really comes in those deeper areas that are not well mapped and characterized. So for example, in 2019, we completed a cruise throughout the entire Dry Tortugas to Florida Keys and identified a new area of mesophotic or middle light reef down at about 160 feet that had only recently been identified with multi-beam bathymetry and had never been seen in person. So that was really interesting and fun. Unfortunately, a lot of our monitoring work has been focused on looking at how corals change. And for the vast majority of reef systems that we study, those changes are for the worse, not the better. But needing to understand how those changes take place and what drives them is really important. And most of that understanding comes from not just observational studies, but controlled experimental studies. So if we think that nutrients might be a problem, we need to test nutrients in a controlled setting. Or if we think turbidity is a problem, we have to test turbidity in a controlled setting. And then to understand how that's actually affecting corals and their symbionts, we use a suite of advanced molecular tools to understand the genetic underpinnings of what's going on and to try to design future work that can help preserve and protect things. So really it's this integration of three very different methods that leads to some innovative research and data products that we can provide to managers. And so we provide those data and recommendations directly to the management agencies. What I've found is that we provide the best data and the best recommendations when we work with the management agencies in the development of our experimental designs and monitoring plans from the outset. That tends to work best. And then lastly, in our role as a university, student training and developing effective outreach is really an important part of the mix to making sure that we have leaders and expertise for the next generation. And really my primary role is to help to develop a sense of innovative leaders that will come up with ideas that are better than all of the ones we have currently. So to give a kind of background on our Florida reefs here, we do have the third longest barrier reef in the world at about 225 miles, stretching from the Dry Tortugas all the way up to Martin County. It's a relatively young reef, geologically speaking. It's only about 5,000 to 7,000 years old. It's arguably the most highly managed reef ecosystem in just about anywhere in terms of number of management units per area. We have Dry Tortugas National Park. We have the Florida Keys National Marine Sanctuary with several different inclusions within the sanctuary. We have John Pennekamp State Park. We have Biscayne National Park. And then we very recently have the Southeast Florida Coral Reef Ecosystem Conservation Area, or you'll sometimes hear me call it the Southeast Florida Coral ECA. So this is a new protected area that was just established in 2018. And management plans for that area are currently under development. So although we have a relatively long reef, here in Florida, we're kind of getting to the margin of what makes for great coral habitat. We still have incursions of low temperature during the winter that make this good but not ideal situations for coral. We also have heavy freshwater influence, both from natural drainage through the Everglades, as well as managed drainage along the East Florida coast that I'll talk about in just a moment. Those things combined mean that we've historically had relatively low coral cover in the past 100 years or so. It's only been about 3 to 13%. And that's in comparison to somewhere like Bonaire that has over 50%, or Flower Garden Banks in the Northwest Gulf that also has over 50%. Unfortunately, we've also seen really dramatic declines in that 3 to 13% from several different disease outbreaks and bleaching events And the most recent one is what I'm going to focus on today. So our research has really been focusing on reef health, the underlying ecology that drives that, and then trying to come up with some measures that we think can lead to conservation or restoration of these systems. I'm going to focus today on some of our work at St. Lucie Inlet, since this is a Martin County focused audience. And again, that's up at the northern end of the Southeast Florida Coral ECA. And it is an area that gets a strong amount of influence from freshwater discharge, both from local runoff as well as Lake Okeechobee discharge through the canal system. So that freshwater influence is a really important driver to the whole Southeast Florida Coral ECA. This is an area that has 9 independent inlets, some of which were natural, many of which were either created or enhanced during the population of Southeast Florida and development of Southeast Florida. And as you can see from all of these different canals that link between Lake Okeechobee and the coast, the whole Lake Okeechobee management system is a really important driver of the freshwater discharges and inputs to our coastal ecosystems here. And that's probably more apparent in Martin County than in any other part of Southeast Florida. So we have issues to deal with in terms of Lake Okeechobee management overall, aging infrastructure that needs to be improved, and kind of a hope to return to historical flows through a number of different management projects related to Everglades restoration. Simultaneously, we have a still rapidly growing Florida population. And that increasing population is going to result in increased infrastructure needs, both in terms of freshwater supply and what to do with the wastewater that's generated. So those are big issues that we are dealing with currently and will need to continue to deal with. One of the bright spots is that we did pass legislation in 2008, the Florida Ocean Outfall Legislation Program, which mandates that all outfalls are going to be stopped by 2025. It also mandates that that 60% of our water will come from reusable sources by 2025 as well. I think that there's going to be some challenges in meeting all the targets by 2025, but several areas are on track, but it does require a large amount of investment at several different county, state, municipal levels. But some areas are doing great. So for example, Delray and the Delray Boynton outfall stopped regular discharges way back in 2009. They still have had to occasionally do emergency discharges when some of their systems get overwhelmed, but regular discharges have now been not occurring there for over 12 years, which is fantastic. So you can see that here in red, we have that reef area for Southeast Florida, and you can see how influences from those inlets and from the discharges are going to impact potentially coral reef health in our area. And again, St. Lucie is right up here at the top, just to point that out. So St. Lucie Reef is probably one of the more interesting locations in the Southeast Florida Reef Tract because it's really at this ecotone transition. You start to get to more temperate weather patterns during certain times of the year at the northern end of Martin County. And so we're really kind of at the northern limit for corals living along the east coast of Florida. There are also corals farther north in this in the Bahamas. There's corals farther north in this in Bermuda, but there's not as many corals that extend north of the St. Lucie Inlet. Only about 4 species of corals persist north of that, and it's just a few small individuals along the coastline. So this is an important resource. Part of being at that ecotone transition means that it gets both temperate and tropical fishes. So there's over 250 known fish species within St. Lucie Inlet State Park, and it's widely used for both commercial and recreational fishing. Kingfish, mackerel, snook right now, for example, are all fished within this resource. There's 2 important corals that kind of historically been dominant there, a brain coral called Pseudodiplora clavosa and a star coral called Montastria cavernosa. So a lot of the work that I'm going to present today focuses on this Montastria species, which is ubiquitous throughout the tropical Western Atlantic, from very shallow reefs all the way down to deep reefs as well. So this entire region really gets impacted heavily by these occasional discharges from St. Lucie Inlet. So some of those discharges are controlled by Lake Okeechobee discharge water, but others are just related to rainfall and runoff into the estuary overall. Here in the September shot up on top, you can really see that very stark boundary between the clear oceanic water that's starting to get sucked in on the north end of the inlet as the tide starts to change, while black water, fresh water, is still continuing to come out the inlet here on the south side. And during particularly bad discharge events, that dark black fresh water can persist up and out towards Bathtub Beach and south along the entirety of St. Lucie Inlet State Park. And we've been able to document salinity as low as 16 parts per thousand on some of the coral reefs within St. Lucie Inlet State Park. For reference, a normal coral reef is happier around like 35 to 37 parts per thousand. So we know that this area is getting potentially exposed to some of these low salinity incidents during discharge events. And the discharge tends to carry with it excess nutrients, excess particulates, darker water color or dissolved organic carbon. And so we wanted to kind of get a sense of what those effects might be on some of the reef ecosystems in St. Lucie Inlet State Park. So part of it was to understand when those discharges occur. And so thankfully there was already some existing infrastructure that we could draw from, both the DB Hydro database managed by South Florida Water Management District, as well as the Indian River Lagoon Observatory, which is run by Dennis Hanasak here at Harbor Branch. So they have land ocean biogeographical observation systems, or LOBOs, that are set up throughout the lagoon. There's actually more than what's even shown here, but this is what was available when we started this study. And then we've also deployed some salinity and temperature sensors out on the reef. And more recently, Kathy Fitzpatrick and some volunteers from Orca have organized putting other sensors out there as well. So there's really good infrastructure for understanding water quality and flow dynamics within the lagoon and in the estuary. But once we get out the inlet, there's a paucity of infrastructure, and that's certainly a need that we need to increase going forward. So in addition to collecting all of those environmental data, we were going out and collecting small tissue biopsies at various different times, both during discharges and during non-discharge times. And we developed a really nice optimized molecular pipeline where every little piece of coral that we sampled was getting split to look at least 5 different things. So we were taking initial samples to look at population genetics, or how these corals are related to others throughout Florida, and gene expression, or how they're responding to the environment around them. Then we would take the rest of the coral tissue off and separate the algal tissue away from the coral tissue, and look at things like the total number of algae within the coral, how much chlorophyll those algae were making, and what type of algae were being produced. And then from the remaining coral fragment, after we took all the tissue off, we could 3D scan it to look at surface area and differences in the morphology of the corals as well. So I'm going to focus today a little bit on some of our gene expression results, and then I'll touch later on some population genetics as well. So I recognize that there's a bit of a complicated chart over here on the right, but essentially what we did was we extracted the RNA, or what genes get turned on under different events within the corals. And what we found was that after studying these over 7 different time points over about 2.5 years, that when corals were the most stressed, that was related to thermal stress and exposure to Lake Okeechobee discharge water. And we did this through both in situ monitoring and a controlled ex situ experiment as well. And not too surprisingly, what we tend to see is that the overall cellular function of corals was negatively impacted when they got too hot or when they were exposed to Lake Okeechobee discharge water. And when you combine the effects of both of those, both being elevated temperature and exposed to Lake Okeechobee discharge water, There seemed to be a synergistic effect. In other words, the two of those things combined were worse than either of them alone. And it just overall impeded cellular function. The corals were not as healthy during the time that they were exposed to these two stressors combined. What was really interesting is that not only were the corals being affected, but their endosymbiotic algae were being affected as well. So we also saw individual effects of thermal stress in discharge water, as well as combined effects on the symbiotic algae. And what it seems is that either the algae know or the corals are forcing the algae to potentially leave, because what gets elevated in terms of expression are things associated with cellular motility and flagella. In other words, the algae are getting ready to get excised or bleached out of the corals and make their way on their own in the water column. So that's a really good indicator that even if these corals weren't visibly bleached, that they were on the cusp of becoming bleached. And this work was just recently published in Frontiers in Marine Science. So we finished that study in 2016, and we continued to monitor for another year. And overall, what I would have said if you asked me in kind of early summer 2017 is that the coral populations that we see in Southeast Florida are really well connected to those in other places throughout Florida. Relative to other similar reefs along the coast, St. Lucie Reef had really high levels of biodiversity, particularly within fishes and other benthic invertebrates. We saw that there were these seasonal responses of the corals, and that they were responding to thermal stress and they were responding to discharge events, but that overall the corals appeared somewhat resilient, that even during some of these major discharge events, only a handful of the corals were dying and the whole reef was staying intact. So I would have said that the corals are persisting and relatively resilient, but they weren't thriving, and they were quite vulnerable based on their low population size and their low growth rates, and the fact that we had never observed them being reproductive anytime within that 7-year span. Then, unfortunately, in 2017, a new disease showed up at St. Lucie Inlet State Park called stony coral tissue loss disease. This is a really distinct disease that has a line of necrosis. So you can see on this slide, you have areas of bare white coral skeleton, and right along that line is where the advancing disease starts to eat away, and literally the coral tissue just starts to slough up and off of each colony. So it's a really rapid tissue loss disease. Corals are quite likely to die entirely if they're affected with this disease. And at least 22 different coral species are known to be susceptible to it in South Florida. So the disease first showed up in Miami, near Port of Miami, and Government Cut shortly after a major dredging project. And there's several different debates about whether or not that dredging project contributed to the original outbreak of disease, but we were not involved in those studies, but I think it's plausible. And then after starting in Miami, what's undeniable is that it then spread geographically both north and south along the Florida Reef Tract over time. What was interesting is that it got to kind of the Palm Beach, Jupiter area in 2015, but then it took a long time to make the jump across to St. Lucie Reef. You know, it appeared to be resilient either through isolation or maybe through the fact that the corals were relatively tough in this dynamic, difficult environment. I had harbored hope briefly that these corals would maybe be so tough they would be resistant to this new disease. But unfortunately, in 2017, the disease did show up. And it's since, unfortunately, as of about 2 weeks ago, spread all the way south to Dry Tortugas National Park as well. And it's really resulted in some pretty massive coral mortalities throughout the entire Florida Reef Tract. If you look at the time series here on the left, you can see that a coral that was perfectly healthy in June of 2017 started to show signs of infection by fall of 2017. And then by the following spring, the entire colony was dead and gone. And this was the case on many different reefs throughout the Keys, where huge, large colonies were lost to this disease. There's been various different responses to that, including interventions to try to treat the corals, which I'll discuss later, as well as rescue efforts to go in and try to get some genetic stock of corals in advance of disease coming in and wiping them out. So we wanted to get a handle on how this new disease was going to be impacting coral reefs in the northern end of the Florida Reef Tract. So we established monitoring sites at St. Lucie Reef, in Jupiter, in West Palm Beach, right off of Breakers, and down in Broward County near Lauderdale by the Sea. And over the course of basically the past 4 years, what we've observed is some pretty significant disease impacts, particularly at St. Lucie and Lauderdale by the Sea, where at any given time, prevalence or the percent of corals infected by the disease was as many as about 1/3 of them. What you see here is that there was kind of these episodic changes over time. And we saw these kind of big blips of disease in the blue bands at St. Lucie Reef. And then we haven't really seen any at St. Lucie Reef since June 2019. And while I'd love to tell you that's a great story because the disease is gone, the truth is the reason we don't see much more disease at St. Lucie Reef is that most of the susceptible coral species have been wiped out. So the Montasteria cavernosa and the Pseudodiploria clivosa, there are still some individuals left, but they are relatively small individuals and not nearly as prolific as they once were. There are other coral species at St. Lucie Reef that have survived and thrived. Things like Parietes, for example, and Parietes asteroides have done quite well. What's been interesting at Lauderdale by the Sea is that there's been kind of a persistent and ongoing infections in those populations further to the south. And overall, Jupiter and West Palm Beach haven't fared too bad since spring of 2019. There probably was some initial infection that we missed because we know it first arrived there in 2015. So we don't have the earliest disease data for those sites in this data set. So to sum that up, what I would say is that there were really kind of devastating impacts in both of these regions. And we used some 3D photogrammetry to understand some of those impacts and tell that story. But watching corals die was incredibly depressing. And so we wanted to come up with a way where we could try to treat the corals in a way that might save coral tissue. And some colleagues at Nova Southeastern University and the Keys Marine Lab, Karen Neely and Cindy Lewis, had been working with a group called Ocean Alchemist to develop a possible treatment. And we were asked to test that. And so we did a controlled test comparing this silicone-based amoxicillin treatment against some chlorinated epoxy, and then 2 different levels of controls. And long story short, what we found was that over an 11-month period with just a single application, this amoxicillin treatment was incredibly effective, more than 95% effective within that span. So it really provides some good hope and some potential alternatives to managing what goes on when a disease comes in and starts to hit a reef. So this is currently kind of the standard that is being used both throughout the Keys and now in the Dry Tortugas, as well as the Virgin Islands, to try to treat SCTLD as it comes into a coral community. So what we don't know is that with this disease coming in and starting to wipe out various different parts of coral populations, what's the bigger impact on coral reefs? How is that going to impact the populations that we have in our systems, how those populations are connected to one another and persist, and then how do we manage those and appropriately plan coral restoration after an event like this? So to try to get at some of those questions, we've taken on a multi-species approach with 3 different coral species and a sponge species, and we've adapted a genotyping pipeline called 2B-RAD that can identify individual base pair differences in the genes of those corals. And what we found is that there is some level of relatedness, but for example, Montasteria cavernosa, the great star coral, has these distinct populations across Florida. And so maintaining local populations is important, but we also need to maintain those upstream populations that may serve as sources of new larvae coming into the system. Understanding these differences in genetics can also be useful for designing restoration strategies. So for example, we may have certain types of Montasteria cavernosa that appear better adapted for life in Southeast Florida as compared to life in and conditions in the Florida Keys. So we may need to outplant different kinds of individuals in those different areas. So this is a big effort in our group. We're also working with FWC, who's leading some genotyping efforts for some other species as well. We're also collaborating with NCOS through NOAA and a group at Lehigh University to expand this into the Northwest Gulf of Mexico also. And again, this is work that's also recently been published for the Florida Keys if you're interested in digging into this more. So our overall approach is going to be to use these several different species, and we've essentially collected samples from almost all of these areas now. In Flower Garden Banks National Marine Sanctuary, off the northern coast of the Yucatan, in Belize. We completed a circumnavigation of Cuba in 2017, where we sampled corals all the way around Cuba. And then we've done a number of different expeditions out to Pulley Ridge, Dry Tortugas, all of the Florida Keys and Southeast Florida. And at least for a few of these species, what we've found is that often areas are connected across very broad geographic space. So we know that mesophotic or deeper corals in Belize are connected to corals on the western end of Cuba and connected to corals in the Florida Keys and Dry Tortugas. So that suggests that in terms of long-term sustainability of our reef systems, we need to think about international reef management and investing in other locations in ways that may benefit us being downstream from them. So to sum up, kind of main take-home messages today, I really want to reinforce the idea that coral reefs are really kind of ecological and economic powerhouses, but they are under threat. When those threats are abated, corals can be pretty resilient and can come back when given the chance. But when several threats are stacked on top of one another repeatedly, disease, climate change, local anthropogenic impacts, it makes it more difficult or less likely that those corals can recover. So abating some of those threats can really go a long way. We know that these direct interventions with antibiotics can help to save corals, but we really are going to have to scale that up to something that's ecologically effective in order to save entire reef systems. So that either means armies of people going out to apply antibiotics or alternative treatments that can be applied more broadly to a reef. Essentially everywhere that we've gone looking for mesophotic reefs, those middle light reefs that are deeper around about 150 feet to 400 feet, we've found them. They're extensive around the entirety of Cuba. They're extensive off of the Florida southern coast and southwest coast. They're extensive across the Northwest Gulf of Mexico. And we're hoping that perhaps these deeper areas may be kind of more isolated from some of the pressures that shallow reefs are facing and may provide a source of recovery for shallow reefs. In other words, a source of larvae that could help to repopulate shallow reef ecosystems when given the chance. We've been involved in either the creation or expansion of several new MPAs across the area and are involved in revised management plans for several of those. But I think that there's really still quite a bit of work to do to secure a true future for corals in Southeast Florida. So to try to get at some of those areas that still need to be addressed, we're going to continue our work with the stony coral tissue loss disease and the intervention techniques to look at how intervention and disease impact gene expression, how it affects the microbiomes of the coral, how both the disease and intervention affect the symbionts, and then how it affects the coral metabolism. So things like lipid development, for example. We have two new projects that have just started this year. One of the interesting things about one of the coral species, Orbicella faviolata, is that in Southeast Florida and in the lower Keys, right next to one another, there can be individuals that are susceptible and individuals that are relatively resilient or resistant to the disease. And there's a large group of us that is really interested in why. So in collaboration with NOAA, University of Miami, and several other partners, we've sampled several of those populations and will continue to do so over time to try to understand what's different about them, what makes certain corals more resistant to disease than others, and can that be a trait that we can select for going forward. We also just completed the largest experimental restoration project in Florida's history. Our team planted out about just under 200 coral colonies at St. Lucie Reef and in Palm Beach. And that's part of a project that planted about 1,000 colonies out overall across the entire Florida Reef Tract. Again, led by FWC with several different partners involved as well. And so what we hope to identify is, number one, can corals that come from different places be successful in alternative environments throughout Florida? And then what is the relative risk of these corals that we plant out in contracting disease and dying? Are we wasting time and effort in doing this, or can it be effective, and how can we improve and optimize that going forward? And then some new areas that we're hoping to move into. We know that we need to understand the impacts of salinity on corals, particularly with regard to Lake Okeechobee management. And so while there's some decent data for the salinity thresholds of corals in other parts of the world, we don't have good salinity threshold data for corals here in Southeast Florida. So we've just recently gotten a green light on funding to try to look at that in partnership with DEP and Martin County. I'm also really interested in the nutrient impacts on SCTLD. So we've done previous research that has shown that excess nutrients in the water can make corals die faster to things like black band disease and yellow band disease. And I'm curious if that's the same for this new stony coral tissue loss disease. So we just got permits this past week to start to conduct some of that work to understand if nutrients are driving some of the SCTLD dynamics that we're seeing in Southeast Florida. And then lastly, if we're going to continue with some of these antibiotic treatments, we also need to understand if antibiotic resistance development is a risk. We're using amoxicillin, which is already present in the system through sewage practices, but we want to make sure that we're not accidentally increasing antibiotic resistance by using this treatment. So that means using enough to be effective, but not too much, essentially. And then some of my overall recommendations going forward that I would say, those of you listening in today can champion and share with those in your various spheres. We know that we've got to support water quality infrastructure improvements in South Florida. We also know that it's relatively expensive, and it's not a technological limitation at this point. It's a will and a financial limitation that we need to overcome in order to meet those targets and achieve those goals. Now that we have this new Southeast Florida Coral Reef ECA, it needs a management plan, and that management plan needs to be implemented. So the box has been created, but there's still not a lot of regulations that are going to determine what can happen within that box and how those regulations will be enforced. So I think that's an important next step. Again, I mentioned this before, but I really think we need to do a revised economic evaluation for coral reefs, particularly here in Florida. I think that that could go a long way into some of the calculus for how decisions are made legislatively and executively in our state. So I would, while that's not my expertise, it's something I would advocate for. And then, you know, we're to the point where Florida's coral reefs have been so impacted that we're going to have to be somewhat proactive about both intervention and restoration. And I think that we need kind of innovative and successful, effective approaches to achieve that. But we have to make sure that we're still doing that with hypothesis-driven, tested research to make sure that the choices that we're making are ones that are long-term going to be successful for our region. And then lastly, I think it's really important that we engage legislators and voters and leaders throughout our communities through storytelling. And that's something that we've started to kind of operationalize here at FAU Harbor Branch, where we literally train our graduate students and interns on how to give presentations of various different lengths to various different audiences to effectively communicate some of the urgency and importance of good water practices and coral reef management in our state. So if you'd like to learn any more about us, here's some contact information for our team. And with that, I'd be very happy to take a few questions. Thank you all.
Thank you so much, Josh. That was a fantastic overview, really at a comprehensive and robust level for our water ambassadors. So I thank you for that. And I thank you for ending on sort of recommendations and strategies for moving forward and on a positive note. And as someone who does outreach and education as their job, I really appreciate sort of your understanding and acknowledgement of how important sort of both the research and the outreach are to the success of, you know, hopefully this restoration of our corals. So we have a number of questions in the chat box, and I'm going to start with Stacy's, and she had the same question I had. So this is going back to your first data slide where you talked about sort of the stressors of both heat and Lake Okeechobee discharges. And she asks, if the data indicate if the stress to corals and the symbiotes were from the composition of the discharge itself or the effect from light reduction from the discharge that's causing the stress.
Great question. So for the data that was collected from the corals that were out on the reef, we can't disentangle those two. They're simultaneously exposed to both what's in the water and the reduced light at the same time. But for the experiment that we conducted in aquaria on land, I can unequivocally say that light was kept constant in those systems. So the only difference was the composition of the water that they were being exposed to and the heat stress treatments. And so at least for those trials, I'm really confident that that was from the composition of the water, not just light. We could do a separate experiment out on the reef using some shading to try to get at that question more. But I think both are probably a factor, but from the ex situ experiment that we did, we were only testing the composition, not the light reduction.
Great, thank you. And as we move on to our next question, most of our participants have found it already, but I did pop up our evaluation poll for you. This is just to let us know what topics you're interested in and how spot on we were. The next question is from Nicholas, who asks that you found that the corals were stressed due to heat, but was there any data collected or information to see if cold had stressed or is a potential stressor to corals in the St. Lucie Inlet?
So just to clarify real quick, there are no corals in St. Lucie Inlet. There's corals in St. Lucie Inlet State Park. I don't want folks to start going for a dive through the inlet hoping to find corals. Probably shouldn't dive in the inlet, period. A little bit dangerous, but no worries. So this area certainly gets thermal stress on both ends. And I would surmise that part of the reason we don't see fertilization success or gamete development success in these corals is because they don't grow enough during a given growing cycle each year to have energy to put towards gamete resources. There's also times of cold water upwelling on these reefs. And we've seen that signal in our temperature data, but that signal was not well resolved in the gene expression data that was associated with that. So while it may impact their growth rate, it didn't necessarily result in overt signs of stress at the cellular level. And then in the lab experiment, we did not test low temperature stress. We only tested ambient temperature against elevated temperature. So I'm not sure about that one. I will say that previous studies have shown that cold stress slows down coral growth rate, and that at some point, essentially corals crash, where they can either, if it gets too cold or if it stays cold for too long of a time, corals can die.
Thank you. And our next question comes from Vincent, who asks whether or not the stony coral tissue loss disease pathogen has been identified.
It has not. So pathogen ID and marine diseases in general is really challenging. So to date, we have about 33 described coral diseases and syndromes. And of those, pathogens have only been identified for, I think, 7 at this point. 6 for sure, maybe 7. There's another one that's being worked on right now. But part of the challenge is because they're in this marine environment, there's bacteria and viruses on everything. So it's very difficult to tease out what the individual pathogen is. There are several different groups that are working on that right now, including our group that is done some microbiome profiling of corals that have and don't have disease, and corals that have been treated with antibiotics and those that have not. And there seem to be some trends. Julie Meyer up at UF has demonstrated that on corals that had SCTLD, there was an increase in kind of groups of bacteria that are commonly associated with pathogens. So things like Vibrio, Roseobacter, things like that. But no one has yet identified or cultured an individual pathogen. And that may be because it's non-culturable. They may not be able to get it to grow by itself in the lab. The other possibility, and we've seen this for other coral diseases like black band, is there may not be a single pathogen. It may be a consortium of bacteria that kind of work together to elicit this pathogenic response within the coral tissue. So there are several folks working on that, but there is not a defined pathogen yet.
Great. And as we're nearing 1 o'clock, we've got one last question for you. So we'll take this question and then just say thank you so much for a fantastic presentation and for joining the Water Ambassadors. So Mike Sipos, who is our Sea Grant colleague over on the Southwest Coast, says, great presentation. I was wondering what the typical ideal water parameters were on Atlantic reefs regarding alkalinity, calcium, nitrate, etc. I know ocean acidification is talked about often, but how greatly has it affected carbonate hardness offshore? And does it fluctuate seasonally?
Those are great questions. And I'll say, quite frankly, that there's not a lot of great OA-related data for a lot of the shallow reef areas in Florida yet. We have a colleague here, Mingxuan Zhang, that's been working on that a bit. What I will say is that my guess for reefs in Southeast Florida, if you look at the trajectories of OA impacts relative to calcium carbonate dynamics within corals, thermal stress is going to be a much bigger problem than ocean acidification stress for Florida corals, at least Florida shallow corals, much earlier on. for deep corals off of Florida, Oculina and Lophelia. As pressure increases, those dynamics shift and the thermal dynamics shift. And so they are potentially at risk for OA the farther you go down. And then in terms of nitrate parameters, there's various different discussions right now about defining nitrate TMDLs. And what we've seen is that it's been difficult to get time series data with enough frequency on reefs to capture spikes as they come out. Often nutrients as they come out of an inlet or a discharge are rapidly being absorbed, so other methods have to be used other than just looking at nitrate concentration in the water. So things like stable isotope analysis, for example. But I would say that it's pretty dynamic around Florida, all of those things, because we're on a reef that has more seasonal variation than most other reefs throughout the Caribbean or the world. All of these factors change a lot more within a season than they might elsewhere, which kind of adds a layer of complexity to everything.
Yeah, and Vincent adds to that. He wonders how, it just moved. Estuarine acidification affects those reefs as well.
That's another great question to ask. I think we don't know much yet about, as estuaries acidify, like how much of a overall impact might that have as it comes out of reefs. That's folks that the water quality modelers need to focus on. That's well outside my realm of expertise.
Well, we are at 1 o'clock. I want to thank you so much for a fantastic presentation. You have a lot of thank yous and excellent presentations in the chat box. So it was very appreciated, not just from us, but from our participants as well. And we hope everyone joins us for the next Water Ambassador program, which is on the 29th, and it'll be on the Florida Healthy Beaches program. So thank you again, Joshua.
Thank you, Lisa. Thank you, Vincent. Thank you, everyone, for taking time today.
Bye, everyone.
Bye.